Video, we will discuss about the introduction, definition, and the general principles of calibration, qualification, and the validation, importance and the scope of validation, types of validation, validation master plan, calibration of pH meter, and the qualification of UV visible. So let's start with the introduction. In pharmaceutical industry. So let's start with the introduction. In pharmaceutical industries, we use specific types of an analytical instruments for the analysis of different types of drugs or pharmaceutical preparations. For this purpose, we need to ensure that those analytical instruments should be calibrated and validated to provide an accurate results without an error. What is the term calibration and validation? So we will discuss about this term calibration, validation, and qualification one by one. First is calibration. In calibration, we check the activity of an instrument by comparison with the standard results, such as pH meter, weighing balance, etc. In the other hand, the validation is an integral part of the quality assurance. In the validation process, we check whether the analytical instrument is properly installed or not, effectively working or not, without an error. In the validation process, we does not compare with the standard results. Third is qualification. Qualification is a part of validation. Qualification is a process through which an instrument works properly and produce an expected results. There are some types of a qualification like design qualification, performance qualification, installation qualification, and operational qualification. Let's discuss these different types of qualification in detail. In this chapter, we need to understand the validation as mentioned by their name. The design qualification is a documented evidence that the premises or utilities of equipment that it is an integral part of the quality assurance, GMP, right? In validation, the installation the instrument is properly working. It ensures that the instrument is properly installed or not, the process, as per the requirement or specification. It confirms that the process has been properly qualification, and under the control documented verification that the instrument is properly operating or not. Batch provide the and the last is the performance qualification, which met the specification. Again, it is also a documented so that it can be that the equipment operates consistently and whether the equipment gives the results within the standard. There are some different definitions which are given by the different agencies. Like according to ISO, the validation is a confirmation by examination and provision of objective evidence that the particular requirement for a specific intended use are fulfilled. According to US FDA, the goal of validation is to establish the documented evidence which provide the high degree of assurance that a particular process will consistently produce a product which meeting its predetermined specification and the quality attributes. As per the European Commission, the validation is an action providing in accordance with the principles of GMP that any procedure, process, equipments, materials, activity or the system actually lead to the expected results. See the importance of validation process. The validation process or validation ensure the great importance for quality assurance and the cost reduction. Second, through validation process, we can identify or we can assure that the product is intended use or produced as per the QSEM guideline, quality, safety and effective guideline. Third, the parameter which can be controlled if we validate the process or the equipment. Fourth, it helps to determine the worst case if we validate the process or equipment during the manufacturing. So it directly or indirectly reduce the cost reduction after the manufacturing or in the testing procedures. Next, Validation helps to investigate the deviation which caused during the process. So we can easily timely correct that particular deviation arrived during the manufacturing process. Next, the deep study and the understanding of the system and equipment are made possible due to validation. Next, the risk of regulatory non-compliance is minimized after the validation. Next, the validation process requires less process control and the finished product testing, because we already discussed or studied the validation process before or during the manufacturing. Next, batch to batch variation is minimized due to validation of the process, system, and equipment. As we discussed in the definition of the validation, that it verifies, or through the validation, every batch should be uniformly produce the results. Next, reduce the production cost of the product. Next, it also increased the production of the manufacturing facilities due to minimizing the rework and the rejections. Next, it also decreased the chances of the failure of the batches. It very effectively produced that the every batch should be provide the effective results. The validation process also ensure the quality of time-bound process optimization, reduction of quality cost, minimal batch failure, and improve the efficacy and productivity of the batch, reduction in rejections. It increased the output. It fewer the complaint about process-related failure. It reduced the testing in process and in finished goods. The more rapid and the reliable startup of the new equipments. It easier maintenance of the equipment, improve the employee awareness of the process, the more rapid automation, and the government regulations. Scope of validation. The validation process required the sufficient infrastructure, including organization, documentation, person, and the finance. It also required the direct involvement of management and the quality assurance person to take the decision freely. Next, the person with appropriate qualification and experience to perform the validation processes. Next, extensive preparation and planning before validation is performed. Next, validation should be performed for new premises, equipment, utilities, and the system, processes, and the procedures at periodic intervals and when major changes have been made. The validation in accordance with the written procedures or protocols. Next, validation over a period of time, at least three consecutive batches to demonstrate the consistency. Next, significant changes should be validated if arrived. Next, the risk assessment approach is used to determine the scope and extent validation needed. Types of validation. So there are different types of validation. First, process validation. Second, cleaning validation. Third, equipment validation. And fourth, analytical method validation. First is the process validation. So process validation is a document evidence which gives the assurance that the process is producing the product as per the predetermined specification and as per the guideline. There are some stages to validate the process. Stage 1, the process design. In this stage, the manufacturing process is designed based on the knowledge gained through development and the scale-up activities. Stage 2, the process qualification. In this stage, the process which designed is evaluated whether it is capable or not to reproducible commercial manufacturing product. And the third is stage 3, Continual process verification is an ongoing assurance during the production that the particular process we designed is in a control stage or a state or not. In the last slide, we had discussed about the stages of the process validation. Next, we will discuss about the types of process validation. First is the prospective validation. The prospective validation is the establishment of documented evidence of what a system does or what is the purpose to do based on the plan. And this validation process conducted before the distribution of the new product. Second is the retrospective validation. It is also establishment of the documented evidence of what a system does and what is the purpose to do based upon the review and the analysis of the existing information. And this retrospective validation performed after the distribution of the product and the product which is already distributed. Third is the concurrent validation. The concurrent validation is the establishment of documented evidence of what a system does and what is the purpose to do with the information generated during the implementation of the system. And the last one is the revalidation. The revalidation is the revalidation of the process, whether there is a change, if there is a change in the packaging, formulation or equipment or process, which may affect or impact on the product effectiveness or the product characteristics. The second type of validation is the cleaning validation. As mentioned by their name, the cleaning validation is used to describe the analytical investigation of the cleaning procedure, whether the equipment or the facility properly or effectively cleaned or not, whether the residual of the manufacturing product properly cleaned or not below the predetermined level. It also ensures the safety and the purity of the product. There are some types of a contamination which directly or indirectly used to determine the cleaning validation. First is the cross-contamination, microbial contamination, contamination by cleaning or sanitizing agent, and the contamination by other agents. The third type of validation is the equipment validation. As per the Food and Drug Administration, May 1987, the action of providing that any equipment works correctly that lead to expected results is the establishment qualification or equipment validation. It is not a single step activity, but we need to perform the different types of activity which may result the equipment validation process. There are some steps which involved in the equipment validation. First, design qualification. Second, installation qualification. Third, operational qualification. And the fourth is the performance qualification. The last one is the analytical method validation. The analytical method validation is the validation method or a process used to confirm that that analytical procedure which used to perform the specific test is suitable or not. There are the different types of procedure which to be validated, like accuracy, precision, repeatability, reproducibility, specificity, linearity, intermediate precision, robustness, and richness. So first is accuracy. Accuracy means the closeness of value obtained by the test sample to the value of the standard. That's it. Second is the precision. So precision is an analytical method in which the value obtained is close to the standard by performing the series of the test. Next is the repeatability. It is as mentioned by their name, the repeatability means the same sample is estimated rapidly by same analytical analyst using same analytical method within the same laboratory by using same instrument. The next is the limit of detection. In this method, we can just estimate the presence of an analyte, but we cannot measure the amount, how much amount of analyte is present in the sample. Next is the specificity. Specificity is an ability or the method in which the presence of an analyte can be detected even when the other components like impurities, degradants may present in the sample. Next is the linearity. The linearity is the ability through which we can estimate or we can produce the test results. Next is the intermediate precision. This intermediate precision is the method in which the variation like equipment variation, analyst variation, etc. can be estimated during the procedure so that the variation can be correct timely and also minimize the number of the experiments. The next is the rigidness. So the rigidness means the reproducibility of the test. It means the test analysis of the same sample is performed under the different conditions like different temperature, different lab, different analyst with the different instrument, etc. Next is the robustness. The robustness is a technique which measures how unaffected it is by little or by changing the little or intentional change in the method or parameters. The next topic in this chapter is the validation master plan. It is a very important document in the good manufacturing practice. In short, we can describe as why, what, where, by whom, who, and when the method is performed. By using this validation master plan, we can reduce the rework, the satisfactory inspections. It also helps in the new drug approval and content of validation master plan, the introduction, methodology, what will be the qualification, how many persons required for the procedure, what will be the schedule, the preventive maintenance, change control, what will be the procedure to follow to perform the analytical method, and the documentation. Next is what is the purpose of that validation master plan. So basically, it can be an overview of the validation operation. Through this, the management can understand how much time will be required and how many persons will be involved in the procedure, what will be the cost of the validation operation. Through the validation master plan, everyone knows about their job, their responsibility, and it also helps the inspector about various approaches to validate the validation activities. Now let's discuss about the calibration. The calibration is the process in which the reading of the equipment is compared with the reading obtained from the reference equipment, which is already calibrated. The next is the calibration of the pH meter. For this purpose, we need to use two-point calibration method, which means we require two types of buffer. One is the pH 7, which is considered as a standard, and the another one is either acidic or the alkaline buffer. We need to assure that both the buffer which we are going to use are at the same temperature. So the first step is turn on the pH meter and wash their tip with the distilled water. Then immerse the electrode first in pH 4 buffer solution and press the standardize button when the reading gets stabilized. Then wash the electrode again with the distilled water and then generally dry it with a tissue paper. Then repeat the procedure for a pH 7 and or pH 10 buffer solutions. Now remove the electrode from the buffer solution and rinse with the distilled water and blot dry using fresh tissue paper. Next topic is qualification of UV visible spectrometer. So as we all know, UV visible spectrometer is a very important instrument which is used in the analysis of various drugs or samples. Uh, we measure the solution's absorbance by or in a UV visible range. The qualification may be defined as a document through which we can identify or we can assure that that particular UV spectrometer or equipment is calibrated or it will correctly installed or the result given by that particular equipment is accurate. For the qualification of UV visible spectrometer, we need to follow some steps. Like first is the design qualification. In the design qualification, we need to assure that we choose the equipment or instrument as per the specification given or suitable for the sample which we need to be measured. The second is the installation qualification. In this step, we install or we follow the instruction as per the guideline. We need to install the instrument in a room where the temperature should be maintained between 15 to 25 degrees centigrade and the relative humidity is between 45 to 80%. The installation site where the instrument is going to install should be away from the dust, corrosive liquid or a gas. After that, we need to remove the instrument from their packaging material and install the computer near the instrument and or load the software into the existing computer. After that, we need to follow or check some parameters at this stage like their specification, their appearance. There should be no visible defect. Next, there is the operational manual should be present or not. All the parts which are required in the procedure should be uh, present or not, as well as the hardware and the software. The next is operational qualification. In this qualification, in this method, we need to check whether the equipment is properly working or not. For this purpose, we need to connect the instrument to the main switch and turn on the instrument. Then we need to follow the instruction which was given by the manual, which is adhere on the equipment. And then we need to check the parameters like wavelength, accuracy, linearity of the absorbance, resolution, wavelength, reproducibility, photometric accuracy, stray light, photometric noise, baseline flatness, and the photometric stabilities. After that, we need to measure the value which is obtained by the instrument and then we check with the standard whether to check whether the equipment or instrument is given the values as per the standard or not. The next is the performance qualification. In the performance qualification, we have to check the overall performance of the instrument. To check the performance qualification, we need to define the performance criteria, which are very important to perform the routine operations. Define the acceptance criteria for these performance criteria selected. Then determine the values which are chosen criteria and check whether it is meet the acceptable limit or standard limit which we set or not. Then decide on the frequency of regular calibration and the performance qualification for the routine use of the instrument. Thank you.